The LyoView software package serves as an accompaniment to our flagship Lyophilisation sensing technology, LyoViz, which leverages through-vial impedance spectroscopy. Processing and interpreting dielectric signals during freeze-drying is often manual, fragmented, and time-consuming. LyoView resolves all of these problems, streamlining the data processing and analysis workflow, making revealing the insights of your dielectric data that much easier. Without LyoView, LyoViz spectra must be processed one channel at a time, producing a number of TViz parameter files, which must then be combined for each channel and merged with the temperature and process log data, which often requires coding due to the asynchronous measurement intervals between dryers and sensing instruments. The main workbench of the app contains the Studies tab bar, where new projects can be created or existing ones opened. Each study page is split into 4 modules, each divided into a narrow collapsible control panel and a larger display window. The first module is the Cycle tab, where the Lyocycle recipe can be defined and edited. Second is the Batch tab, where the vial array is defined and populated. Next is the Dataset tab, where datasets are imported. And finally, the Parameter module, where the processed and merged data can be analysed and exported. In the Control panel of the Cycle tab, we have some fields to provide information about the study, such as the researcher and the location where it was carried out. There's also a General Notes field to provide additional information and comments. The main window is where the Lyocycle is visualized. Steps can be added, removed, and edited from the Row Edit widget in the Control panel, which updates the tabular view and plot of the cycle in the display pane. Moving to the Batch tab, we can define the physical setup of the vial array. The left panel allows us to define the details of the array itself, such as the number of rows or columns, which updates the central display, as well as the vial type, fill height, and TViz vial electrode height, which updates the sensor vial diagram on the bottom right. The vial type can be selected from standard vial dimensions, or alternatively, uniquely defined by selecting custom and editing the dimension fields in the presented form. The form on the right of the page is reserved for defining information about individual vials, which can be selected by interacting with the vial array. The information defined here updates the various display modes of the array. For example, the positions of the sensor vials, selecting between null, thermocouple, and TViz sensors, or the initial and final masses of the vials. By toggling the auto-iterate checkbox, masses can be rapidly populated by pressing Enter after each value input, which updates the color map in the vial array. Once the information has been populated, both the array and sensor vial diagrams can be exported to an image file using the export buttons at the bottom of the page. After a cycle is finished and data has been collected, the respective datasets can be imported into the software from the dataset. This can be done by either selecting specific files or using the bulk import, which searches through files within a folder location and imports and extracts all valid data formats. Imported files are processed and their metadata is compared with a series of pre-configured schema, which allows parameters to be extracted accordingly for each dataset type. These are automatically computed and passed to the parameter module where they are displayed. Plots are split into 2 domains: the temporal domain on the left, where the time series data are plotted, and the spectral domain on the right, where the real and imaginary capacitances are plotted against logarithmic frequency. Spectra can be displayed by clicking on different points in the temporal plots, which marks a time point and extracts and displays the dielectric profile at that point in the spectral plots, as well as inferring all of the data values at that time and outputting them in the values window on the left. Visualization can be manipulated in several ways to display the parameters and region of interest. The plot region can be scaled and panned using scroll and drag interactions with the plot area and axes. You can also define specific regions by either stage, step, or specified range. The axis can then be auto-scaled to fit all the data again, using the auto-scale button in the lower left corner of the plots. As well as absorbing the parameter extraction and plotting functionality of its predecessor, the incorporation of the cycle definition means that cycle recipe data, along with TVIS thermocouple data, can also be included in dataset exports through spline interpolation at each spectra timestamp. LyoView takes the cumbersome task of processing all your datasets after every cycle and does it all automatically in just moments, condensing them all into a simple project file. Save some time and keep your project folders tidy. LyoView. If you're interested to hear more about the insights that through-vial impedance spectroscopy can offer and how LyoView can make your research easier, contact info@LyosenZ.com.